Kingdom, 11th class Biology, Chapter 3, Plant Kingdom. The main topics are algae, bryophytes, pteridophytes, gymnosperms and angiosperms, which are the basic divisions under the plant kingdom. In the previous chapter of the biological classification, we looked at the broad classification of living organisms and the system proposed by Whittaker 1969, wherein he suggested 5 kingdom classification: Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal with further classification within kingdom Plantae, popularly known as Plantae kingdom, plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and the members of Monera and Protista having cell walls have now been excluded from kingdom Plantae, though earlier classification placed them in the same kingdom. The cyanobacteria, that are also referred to as blue-green algae, are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Let us look at the classification within angiosperms to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number and shapes of leaves, etc. They were based mainly on vegetative characters or on the Andricium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on few characters. Basically, before and all, we placed many of the Protistas having basically cell wall was a classification, like was a property to be placed under plant kingdom, but which is not true, because fungi have cell wall, Protista have cell wall, and they do not come under the kingdom of Plantae. And before, the first system of classification used for plants used only gross superficial morphological characters. What these characters they use for classification is this habit, color, number and shape of leaves. They were based mainly on vegetative characters. Vegetative means like the morphological characters, and on the Andricium structure. This system was given by Linnaeus. Such system was artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. The artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable. They gave basically the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. Okay, let's read that again. Basically, the gross morphological based on classification on vegetative character or gross morphological character and based on structure of Andricium, such system was called as artificial system. And through this system, they separated few closely related species due to some special characteristics, and this artificial system gave equal weightage to both sexual and as well as vegetative characteristics. Basically, they divided the plants or not animals, plants only based on the vegetative characteristics only, not vegetative characteristics. Like they give equal for sexual and vegetative characteristics, but they're saying it is not acceptable because we know that like vegetative characteristics easily get affected by the surroundings, environment. Because of such a reason, they claim that the new system should have more weightage given towards the sexual characteristics, because vegetative characteristics are more easily affected by the environment. As against this, natural classification systems developed which were based on natural affinities among the organisms. So against this artificial classification, natural classification systems developed. Okay, this is a new system. Basically, the first was a artificial system. This system used gross morphological characters and structure of Andricium given by Linnaeus through which they separated few closely related species of plants. But we have limitations of artificial system. They gave equal weightage to both sexual and vegetative characters. This is for classifying. The above is unacceptable because vegetative characters are influenced more easily by the surroundings. Artificial system used gross morphological characters, structure of Andricium, through which they separated few closely related species of plants. But because they gave equal weightage to both sexual and vegetative characters, this is unacceptable because uh, vegetative characters are easily experienced, influenced by the surroundings. And against this, natural classification systems developed, which were based on natural affinities among the organism and not only the external features. Okay, but also internal features which were based on natural affinities of the organisms and consider not only external features but also internal features like ultrastructure, anatomy, embryology, phytochemistry. Such a classification for flowering plant was given by George Bentham and Joseph Dalton Hooker. The second was natural classification systems. First is artificial classification system. Second is natural classification system. Natural classification system, here basically they used affinity in organisms. They used not only external structures, morphology, but also anatomy, ultrastructure, embryology, phytochemistry, etc. This was given by George Bentham and Joseph Dalton Hooker. George Bentham and Joseph Dalton Hooker. This is the second type of classification. Now let's come to the third type of classification. Now at present, phylogenetic classification systems based on evolutionary relationship between various organisms are acceptable. This assumes that organisms belonging to the same taxa have common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is equal importance. In the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information. The next is cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses chemical constituents of the plants to resolve confusions, are also used by taxonomists these days. Let's read that again. Now at present, phylogenetic classification systems based on evolutionary relationships of various organisms, these are acceptable. Phylogenetic classification systems based on evolutionary relationship between various organisms, these are acceptable. Phylogenetic relationship meaning evolutionary relationship between various organisms. This they are saying is acceptable. This assumes that the organisms belonging to same taxa have a common ancestor. We now use information from basically the first two statements are phylogenetic classification system are based on evolutionary relationships of different organisms, and this is acceptable. This assumes that the second statement is this assumes that organisms belonging to same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. Basically, the phylogenetic classification, that is one system where we use evolutionary relationships to determine the classification. That is one thing. And we also use, like when we have a problem where we are not able to determine the group, then we use various other classifications as well. I mean various other sources as well for classification determination, like where we need to classify that or how we need to classify that organisms. We use other sources too. That is what they said in the third statement. These become more important when there is no supporting fossil evidence. When we can't clearly say this plant has this ancestor which is not supported by fossil evidence, then we can use other sources, of which numerical taxonomy, this is one category or one other source which we can use, which is now easily carried out using computers, is carried out by computers based on all observable characteristics. All the observable characteristics of a plant we take, and number and codes are assigned to all the characters and the data are then processed. Basically, they will assign special codes and numbers to these specific plants and all their observable characteristics, and each characteristic will have a special code. Then they will match up in this way, like when we give special code for all the characteristics. In this way, each character is given equal importance, and there's at the same time hundreds of characteristics can be considered. That is one way. And then another way is cytotaxonomy, that is based on the cell information. Cyto meaning cell, and that is based on cytological information. The cytological information may be chromosome number, structure, behavior, and the next is chemotaxonomy, that uses chemical constituents of plants to resolve confusions, and these are also used by the taxonomist nowadays. Now, phylogenetic classification. Evolutionary based on evolutionary relationships. Based on evolutionary relationship of organisms meaning, if we classify based on this, the organisms under a taxa are having a common ancestor. This is the two statements on the phylogenetic classification. We use numerous other sources too for classification, like one, it is numerical classification, taxonomy. This is giving specific number and code for all observable characters in an organism doing this, we consider hundreds of different, not organisms, characters, and can classify by matching the characters. Now done through computers. This is numerical taxonomy. Then next we can say chemical taxonomy with cytotaxonomy. cytotaxonomy, observe the cell cytology like chromosomal number, structure, behavior, etc. Then we have chemotaxonomy. This is a cool coloring. No, this seems weird. Anyways, chemotaxonomy, it is based on various, they gave chemical constituents, of plants. This is the classification, where first artificial classification we saw it used gross morphological characters, structure of androecium, and they gave equal weightage for both vegetative and sexual reproduction characters, and that is the limitations, and this is unacceptable because vegetative characters are easily influenced by the surroundings. Then came the natural classification system given by George Bentham and I think Joseph Dalton Hooker. This is basically, they used affinity of organisms, they used special not only morphological, they used anatomy, ultrastructure, embryology, then they used phytochemistry, etc. These characters to determine the classification of the organisms. Then came our phylogenetic classification. This is based on evolutionary relationships of organisms. Based on evolutionary relationship of organisms, we classify them, meaning the organisms in the same taxa have a common ancestor. And we use numerous other sources too for classification, like numerical taxonomy, giving specific number and code for all observable characters in organisms and matching them using a computer. Then we have cytotaxonomy, which we use the cell structures like cytology, basically number of chromosomes, structure, behavior, etc. Then we have chemotaxonomy based on the various chemical constituents of the plants. That is the classification. The first main topic is algae, which is Thallophyta. The division is called Thallophyta, having a thallus-like body. Saying the first topic, algae. Algae are chlorophyll bearing, simple thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichen, and animals. Example on the sloth bear. Basically, they are saying algae are chlorophyll bearing, simple, thalloid, autotrophic, they can prepare their own food, and mostly freshwater and marine organisms. They also occur in moist stones, soils, and wood, and in association mutual relationship with fungi like lichen, and in case of animals present on the sloth bear. The form and size of algae is highly variable. The size of algae and the form of algae is highly variable, ranging from colonial forms like volvox and filamentous forms like Eulothrix and Spirogyra. Basically, the form and size of algae is highly variable. This can be colonial forms like volvox and filamentous forms like Eulothrix and Spirogyra. 3.1 figure. This is volvox. This is a colony. and this is, I think, uh, it is Eulothrix, I believe. No, it is. Wait, yeah, yes, yes. It is. This is Volvox, and this is Eulothrix. This is all. This is comes under green algae. A few of the marine forms, such as kelps, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being zoospores. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large and non-motile static female gamete and a smaller motile male gamete is termed as oogamous. This can be seen in Volvox and Fucus. Algae are useful to man in variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of the food cycles of all aquatic animals. Many species of Porifera, Laminaria, Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce a large amount of hydrocolloids, water holding substances, that is algin, brown algae, and carragin, red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyaceae, Phaeophyaceae, Rhodophyaceae. The green, brown and red algae. Okay, let's read the two paragraphs once more. Algae are chlorophyll bearing, simple, thalloid, autotrophic and largely aquatic organisms. They occur in colonies. They occur in both water as well as moist stones, soils, wood. Association with fungi as lichens and animals, example on sloth bear. They occur as colonies and colonial forms like in Volvox, and filamentous forms like Eulothrix and Spirogyra. And a few of the marine forms such as kelp forms massive plant bodies. And reproduction of algae by vegetative means is through fragmentation, where algae divides to give rise to a thallus which germinates into a plant body. And asexual reproduction is by production of different types of spores. The most common is being zoospores. They are flagellated, motile, and on germination give rise to new plants. Basically, they produce spores, asexual spores called zoospores, which are motile, which have flagella, and these spores germinate to give rise to a plant body. Sexual reproduction takes place through fusion of two gametes. There is also sexual reproduction in this algae, and this is through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, non-motile, but in similar in size as in Spirogyra. In Eulothrix, we see both the gametes are similar in size and have flagella in Eulothrix, and In Spirogyra, both the gametes are similar in size, but both are not motile, non-flagellated. In such a case, both are isogamous gametes, and in case of anisogamous gametes, isogamous is when in Eulothrix and Spirogyra, in Eulothrix they are motile, in Spirogyra they are non-motile. And this kind of equal gametes is called isogamous. Fusion of two gametes dissimilar in size, as in Euderina, as is termed as anisogamous. In Euderina, the fusion of gametes, the gamete sizes is unequal. That is why we call it as unisogamous. And fusion between one large non-motile and female gamete and a smaller motile male gamete is termed as oogamous. This can be seen in volvox and fucus. Algae, simple thalloid autotrophic organisms, autotrophic organisms, largely aquatic, marine and freshwater, found also on moist stones, wood, as lichen, and on animals, sloth bear as an example, and can exist as one colonial form. Example is volvox. Next, second, filamentous form as filamentous. Example, it is Spirogyra and Eulothrix. Example, volvox, Spirogyra and Eulothrix. This is the forms which it can exist as. And the next topic was some of their current. Okay, they gave an example of, and kelp has a very large meters of plant body. And then comes the reproduction. A reproduction in algae. A reproduction in algae is, first one is vegetative. Mainly through fragmentation, where body divides to give rise to a new plant body or new thallus. This is the vegetative method. In case of 2, it is asexual reproduction. Asexual reproduction. This is through zoospores, motile, which germinate to give rise to new plants. Then we have sexual reproduction. This reproduction, it is through, I think they didn't give examples for asexual and vegetative here. Yeah, they didn't give. They did give that those spores are flagellated. Sexual reproduction is again divided into 3 topics, which is a isogamous. Example, we have Eulothrix and Spirogyra. Eulothrix and Spirogyra. Basically, Eulothrix and Spirogyra, right? Yes. Yeah. Isogamous, to motile in case of Eulothrix, or non-motile. This sexual reproduction is through fusion of gametes. To motile Eulothrix, or non-motile, it's not under brackets. To non-motile in case of Spirogyra, identical gametes fuse. This is under the isogamous. B, anisogamous. Two non-identical gametes fuse. This is anisogamous. Example, here on the right. Example, it is Euderina. And in case of anisogamous, I mean in case of oogamous, example is, I believe it was Volvox and Fucus. Where a motile and small male gamete fuses with a large and non-motile female gamete. There is a sexual reproduction Next, I think the topic is uses. Uses. Next is algae are useful in variety of ways. At least half of the total carbon dioxide fixation in Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are paramount importance as primary energy producers, energy compounds which form the basis of food cycles of all aquatic animals. Many species of Porifera, Laminaria, and Sargassum are among the 70 species of marine algae used as food chain. Certain marine brown, okay, fine. That is the uses. Basically, all our uses here. Uses of algae is basically first one, fix an approx of 50% of all carbon dioxide of the atmosphere. That is the first use. Second, up to 70 species of algae, including Porifera, Sargassum, I think it was Sargassum, right? Yes, Sargassum and Laminaria are used as a primary food source for aquatic animals. Then they provide O2 for the surrounding environment. Then the uses are and commercial users are certain red algae, certain brown and red algae. This is certain marine brown and red algae are produce large amounts of hydrocolloids, water holding substances. Large amounts of hydrocolloids, water holding substances. Example, algin for brown algae. Algin produces, brown algae, a certain species of brown algae produces algin, which is a water holding substance, these are substances are used commercially. Algin and carrageen, red algae, produced by Then there's agar as well. From Gelidium gracilaria are used to grow microbes in preparation of ice creams and jellies. Agar used prepared by certain red algae of Gelidium and gracilaria used in preparing microbe cultures, preparing jellies and ice cream, etc. 6. Chlorella unicellular algae, which is rich in nutrients, basically proteins, and proteins is taken is tablets for space travelers. I think that is with the algae paragraphs. Three main, this all the information from the second half of the page to the fourth half of the page, basically, which is saying about the various forms of algae, which are simple thalloid autotrophic organisms. These are largely aquatic, marine and fresh, found on moist stones, wood as lichen. On animals, sloth bear can exist as colonial forms. Example, volvox can be filamentous, Spirogyra and Eulothrix. Kelp has a very large plant body. Reproduction in algae, vegetative mainly through fragmentation, where body divides to give rise to a new thallus. Asexual, this is through zoospores, which are flagellated spores which are motile, which germinate to give rise to new plants. And sexual reproduction is fusion of gametes. Isogamous, example Eulothrix and Spirogyra. Two motile Eulothrix or non-motile Spirogyra, identical gametes fuse. Anisogamous, example is Eudorina. Two non-identical gametes fuse. Oogamous, example is Volvox and Fucus, where a motile and small male gamete fuses with a large and non-motile female gamete. Uses of algae fix an approximation of 50% of all CO2 of the atmosphere. Up to 70% of algae, including Porifera, Sargassum and Laminaria, are used as primary food source for aquatic animals. They provide O2 for the surrounding environment. Certain marine brown and red algae produce large amounts of hydrocolloids, water holding substances. Example is algin produced by brown algae and carrageen produced by red algae. Agar prepared by certain red algae of Gelidium and Gracilaria, used in preparing microbe culture, preparing jellies and ice creams, etc. Chlorella unicellular algae, which is rich in nutrients and proteins, is taken as tablets for space travelers. Now starting with green algae, which is chlorophyllaceae. It's better to write normal. GAC. Basically green algae. Let's see. Chlorophyllaceae. The members of Chlorophyllaceae are commonly called as green algae. The plant body may be unicellular, colonial, or filamentous. Okay, information is they can be unicellular, colonial, or as filamentous, as we saw in Eulothrix and Spirogyra. Unicellular, as we saw in Chlorella. Colonial, as we saw in Volvox. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments localized in different definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. We see a ribbon-shaped chloroplast in case of Spirogyra. When we see Spirogyra, we see a ribbon-shaped chloroplast. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids are storage bodies. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type of formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Chara. Okay, fine. Let's read that again. The members of Chlorophyllaceae are called green algae. Okay, fine. The plant body can be unicellular, colonial, or filamentous. They are grass green in color due to domination of pigments of chlorophyll A and chlorophyll B. And these pigments are localized in definite chloroplasts. The chloroplasts may be discoid, plate-like, reticulate shaped, cup shaped, spiral, or ribbon shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in chloroplasts. Pyrenoids contain protein besides starch. Basically, first information, they are green algae. They are called green algae. They can be unicellular, colonial, or filamentous, and they are green in color due to presence of or domination of chlorophyll A and chlorophyll B. And they have chloroplasts which are localized, which have different kinds of shapes. 4 types of information. And the fifth information, they have pyrenoids. They have special storage bodies called pyrenoids which store both protein and starch. And the next point is they store not only in the form of pyrenoids, some store in the form of oil droplets as well. Next information is the inner cell layer is made up of cellulose. They have rigid cell wall, and the inner cell layer is made of cellulose and outer is made of pectose. And reproduction is in the next paragraph. Vegetative reproduction is by fragmentation. Okay. Asexual is by flagellated zoospores. Okay. And these zoospores are produced by zoosporangia. The sexual reproduction shows considerable variation in isogamous, anisogamous, and oogamous. Some commonly found green algae. Then the next information is examples, which are Chara, Volvox, Eulothrix, Spirogyra, and Chlamydomonas. These are the basic types of information. The first information is called green algae. The second information is maybe colonial, filamentous. Colonial is Volvox, filamentous Spirogyra and Eulothrix, or filamentous and unicellular in case of Chlorella. Unicellular. And next information, grass green in color due to chlorophyll A and B pigments domination basically, and the pigments enclosed in definite chloroplasts of different shapes which may be of a ribbon like, discoid, plate shaped, reticulate, etc. Next information is they store starch and protein in form of pyrenoids. And next information, some also store food in form of oil droplets. Next piece of information I think comes from the reproduction part. Let's see. Okay, it's actually about, I think we didn't miss any information. This can be one or more and they have a cell wall, green algae usually have a rigid cell wall made of cellulose inner wall and pectose outer wall. This is the body types, and as for reproduction, a vegetative, fragmentation, asexual through flagellated zoospores produced in zoosporangia. Next, C. Sexual reproduction is through isogamous, anisogamous, or oogamous. Examples are Chara, Volvox, Spirogyra, then Eulothrix, Chlamydomonas, and that's pretty much all, etc. I think we didn't miss any information here. Yes, that is with chloro 5Se. Next is 5O5Se. Brown algae. So let's use a brown pen. There doesn't seem to be brown, so let's use orange. 5O5Se. There should be an O here. Brown algae. Next is 5O5Se. The members of 5O5Se or brown algae are found primarily in marine habitats. And one more thing, in chloro 5Se is found in freshwater mediums. Found mostly in freshwater mediums. And 5O5Se is found mostly in, in both fresh and salt water, but little more in marine water. And Rhodo 5Se is mostly found in marine water only. In marine water. They show great variation in size and form. They show very much variation in size and form. They range from simple branched filamentous forms, ectocarpus, to profusely branched forms represented as by kelps, which may reach to a height of hundreds of meters. They possess chlorophyll A and C, carotenoids, xanthophylls. They vary in color from olive green to various shades of brown, depending upon the amount of xanthophylл pigment, fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating called algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to a substratum by a holdfast and has a stalk, the stipe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction is most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium, oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are ectocarpus, diota, laminaria, sargassum, and fucus. Let's read the paragraph again. Members of 5O5Se or brown algae are found primarily in marine habitats. Basically found, first information is found in marine habitats. Okay, they show the second information is they show variation in size and form. They range from simple branched filamentous forms, which like example is ectocarpus, and to profusely branched, which is example as kelps, which may reach reach to height of 100 meters. Basically, the second information is basically size and form. In ectocarpus, we see simple and profusely branched structure, whereas in kelps, we see hundreds of meters of long or tall plant body. And the third information is they possess the pigment information. They possess A, C, chlorophyll A and C. They possess carotenoids and xanthophylls, and they vary in color from olive green to various shades of brown depending on the amount of xanthophyll pigment, which is fucoxanthin, present in it. This is the third information of pigment information. And fourth information is food, storage of food. In chlorophyll we saw pyronoid, or in form of basically starch, but in here we see food is stored in complex carbohydrates, which may be in the form of laminarin or mannitol. This is the fifth or fourth information. Next information, the fifth information, is basically the cell wall. The vegetative cells have a cellulosic cell wall similar to chlorophyacin members, but they have a gelatinous layer of algin covering them. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. Basically, they are saying the next information after the cell wall is the cellulosic cell wall is the cytology. Basically, it's not completely cytology. It's the basic cell blueprint, which has protoplast, which has plastids, basically chloroplast, chromoplast, etc. And there is a centrally located vacuole and a nucleus. In plants, we know there is a centrally located large vacuole which occupies most of the cell space, and there is also a nucleus. Next information, and the plant body is usually attached to a substratum, basically a base, by a hold fast, and this structure has a stalk and the stipe and leaf like photosynthetic organ called frond. Basically, the structure is if this is a substratum, there is a hold fast, there is a stalk, a stipe, and a leaf like photosynthetic organ. Frond, stipe, and hold fast. Hold fast helps in addition to the substratum. This is the substratum, which is the base. Hold fast, there is a stipe, stalk like stipe, and there is a leaf like photosynthetic organ called the frond. That is the structural information. Then there is the reproduction information. Vegetative reproduction is same as chlorophyacin, which is through fragmentation. Asexual reproduction is in brown algae is through biflagellate zoospores that are pear-shaped and have unequal laterally attached flagella. It is like euglena, where we have unequal flagella, but here we have a pear-shaped zoospores, and the zoospores have laterally attached unequal flagella. And next is sexual reproduction. After asexual reproduction, we have sexual reproduction here, which may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within oogonium for oogamous species, and the gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are ectocarpus, dictyota, and laminaria, sargassum, and fucus. Fine. Basically, they are saying the sexual reproduction may be iso, aniso, or oogamous, and union of gametes. The next information, the place of fusion is either water or in the oogonium for oogamous species, and the gametes are pear-shaped, the same as zoospores, which are also pear-shaped. And here the gametes, same as zoospores, in the Phaeophyaceae, are pear-shaped, have two laterally attached unequal flagella. This is called a pyriform. The common forms are ectocarpus, dictyota, laminaria, sargassum, and fucus. Okay. First information was found primarily in marine habitats. That is written. Next information can be can show many forms like one simple and profusely branched in case of branched. Example is, I think it was ectocarpus. Then it might be hundreds of meters in height. Example, kelps. This is the second information. Next, the pigment information. They possess chlorophyll A and C, carotenoids, carrot color, carrot, orange or brown color. So brown color is also 5 of I see. Xanthophyll, fucoxanthin, etc. The concentration, the amount of pigments determines its color, which can be from olive green to brown shades. Then the next information, I think, is storage of food material. Food is stored as complex carbohydrates such as mannitol and laminarin. As mannitol, and this is the information about food storage. Then the next is the vegetative cell has cellular, is cell wall and the cell structures. Cell is having a cellulosic cell wall covered by a gelatinous layer of algin and there is cell has plastids centrally large vacuole nucleus and other organelles. Then about the organism structure, the plant body has a hold fast to adhere to the substratum. Then a stalk like stipe and a leafy photosynthetic frond. This is the body structure. And then comes to reproduction, which is fragmentation, asexual is biflagellate. Okay, fine. A reproduction first for, in case of it is, let's take vegetative, it is fragmentation, same as chlorophyllaceae. B, asexual through biflagellate zoospores, through biflagellate zoospores, which are pear shaped, through biflagellated zoospores, which are pear shaped. I think that is all for the yes, they have having two laterally attached, having two unequal laterally attached flagella. This is similar for the sexual reproduction as well. The gametes are like that. Sexual reproduction is through both all iso, aniso and oogamous is seen. Fusion may be in water or oogonium for oogamous species. And the next point was, I think it was, the gametes are biflagellate, pyriform, pear shaped, Gametes are biflagellate, pyriform, pear-shaped, and have lateral flagella. This is this is the end of the 5 of ISC. Actually, there is more for examples, which is ectocarpus, dictyota, laminaria, sargassum, and fucus. Examples are dictyota, sargassum, laminaria, fucus, and ectocarpus was simple, profuse, branched. Laminaria, it was seen that it was one of the species. Sargassum, laminaria was seen to be species which were used as marine food. Ectocarpus, simple and profuse, which we saw in kelps, and kelp also we can take an example. DSLFE. Okay, fine. This is for Phyophysi, which is basically mostly found in marine water. Can show forms like simple profusely branched ectocarpus, hundreds of meters of height kelps. They possess chlorophyll A, C, carotenoids, xanthophylls, fucoxanthins, etc. The amount of pigments determine its color, which can be from olive green to brown shades. Food is stored as complex carbohydrates of mannitol and laminarin. Cell having a cellulosic cell wall covered by a gelatinous layer of algin. Maybe this is the reason they can produce the hydrocolloid of algin, which are extracted from the cell walls. And in case of chlorophyllaceae cell wall, the outer cell wall was pectose, where the inner cell wall was made of cellulose. And here cell has plastid, centrally large vacuole, nucleus, and other organelles. The plant body has a holdfast to adhere to the substratum and a stalk-like stipe and a leafy photosynthetic frond. This is the structure. Reproduction, vegetative fragmentation, asexual, it is through biflagellate zoospores, which are having two unequal laterally attached flagella, which are pear-shaped. Gametes of sexual reproduction are also same, and they can be iso, aniso, and oogamous, and fusion may be in water or oogonium for oogamous species. Examples are ectocarpus, dictyota, laminaria, sargassum, kelp, fucus, etc. Next is rhodophyacea. Basically red algae. That seems like a D. Okay, fine. Mostly marine. Next point is the rhodophyacea members. Let's see the diagram for brown algae, which we have here. Green, this is volvox, this is eulopthrix, this is the frond like structure which we see, stipe, hold fast. Here as well, hold fast. Stipe is this one, and this is the frond, midrib, and air bladders. This is the stipe, this is the frond, and this is the hold fast here as well. Here the species are one laminaria, fucus, and dictyota. Laminaria, fucus, and dictyota. Fucus is like this. Laminaria is a single leaf like structure, where fucus is 3 leaves and dictyota is many leaf like structures of the frond. There is presence of air bladders as well, and There is also a midrib here in the algae. Okay, let's see the rhodophyceae. Members of rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Phycoplant erythrin, erythrocytes, red blood cells. Phycoplant erythrin, red. So red pigment in their body. Majority of the red algae are marine with greater concentration found in warmer areas. Majority of the red algae are marine with greater concentration found in warmer areas. They gave, okay fine, let's read first. They occur in both well lighted areas close to the surface, shallow areas, and also great depths in oceans where no light penetrates at all. The red thallus of the most red algae are multicellular. Basically the body is multicellular, most of them. Some of them have a complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. Okay, this is a statement to be remembered. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by they reproduce asexually by non-motile spores. They reproduce asexually by non-motile spores. In case of Phaeophyceae and Chlorophyceae, we saw zoospores, motile spores, and in case of Phaeophyceae, we saw pear-shaped biflagellate spores, motile spores. But here in Rhodophyceae, we see non-motile spores for asexual reproduction, and sexually also non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. Okay, before we saw Phaeophyceae and Chlorophyceae, it can be iso, aniso, or oogamous, but here it is strictly oogamous, and this reproduction is oogamous and it's accompanied by complex post-fertilization developments. There are not much information here. The common members are Polysiphonia, Porphyra, Gracilaria and Gelidium used for agar, Porphyry and aquatic species of used in food cycle, primary like a grass. Polysiphonia is the first time being mentioned here. Okay, let's read from the informations again from the start. Rhodophyceae, the members of Rhodophyceae are called red algae because the predominant of the red pigment, this pigment is the phycoerythrin in their body. Majority of the red algae are, then they should also give that there is also a chlorophyll A and D pigment here, and in their body majority of the red algae are with greater concentration found in warmer areas. They occur in both lighted regions close to surface of water and also in great depths. Basically the first information called red algae. Second information, pigments of the body, which is the color of the body is because concentration of phycoerythrin or phycoerythrin, and next is chlorophyll A and D, and they can be, the next is the next information, they can be found in warmer areas of water bodies, and they can also be found in shallow areas as well as extreme depths of the oceans where light barely penetrates. And the red thallus is mostly multicellular. Some of them have a complex body organization. The next information is food storage. It is in the form of Floridian starch. In the Chlorophyceae members, we saw it is stored as starch. In the Phaeophyceae, we saw it is stored as mannitol and laminarin. In case of Rhodophyceae, it is stored in the form of Floridian starch. Yes, that is one of the information. And this Floridian starch, which is very similar in structure to amylopectin and glycogen. Glycogen is the food storage in case of animals. In animals, yes, that is the case. Basically, food is stored in as in glycogen in animals, and amylopectin is one more compound. This is a statement: food is stored as protein in starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by, basically there are non-motile and oogamous. The vegetative reproduction, which is very common in all three of the algae, which is by fragmentation, and asexual is non-motile spores, and sexually also non-motile gametes and oogamous, and the sexual reproduction is oogamous and accompanied by complex post-fertilization events. The common members are Polysiphonia and Porifera, Gracilaria and Gelidium. Okay, let's write the information. Pigments found are phycoerythrin, give body red color, B chlorophyll, A and D. Next information they gave was we can find red algae in warmer areas of water, water bodies. They can be found in shallow regions and great depths of the ocean. They can be this is a finding where we can find the algae, and mostly they are mostly multicellular, mostly multicellular organisms. I'll be back. Okay, mostly multicellular organisms. Food stored as Floridian starch. Yes, mostly multicellular organisms with some having complex body structure. Food is stored as Floridian starch. I'll write it again. Floridian starch. Food is stored as Floridian starch, which note point is Floridian starch is similar in structure to amylopectin and glycogen. Food storage in animals. In animals, in food storage in animals. Okay, glycogen and amylopectin. And the next, I think, it is reproduction. Very similar. Okay, and red algae. Okay, reproduction. The next topic is reproduction. Reproduction. Vegetative reproduction is through fragmentation. Asexual reproduction is through non-motile spores. I think we call it as aplanospores. Then we have sexual reproduction, which is non-motile gametes fuse to give non-motile gametes fuse to give actually there is no give, they fuse to form the new organism. There's just bringing in new genetic material and genetic variation. That is why sexual reproduction is actually preferred. Non-motile gametes fuse, and it is mostly or all oogamous followed by complex developmental stages. Sexual reproduction, non-motile gametes fuse is mostly or all oogamous only, followed by complex developmental stages. Example is porifera, porifera, then Polysiphonia, Gracilaria and Gelidium. Etc. Where we saw porifera in the aquatic species, like the food of the aquatic species, the 70 species, one of the 70 species, and Gracilaria and Gelidium help in the formation of agar. This is the end of Road of ISC. Basically, let's see once more. Basically, Road of ISC is mostly marine organisms. Pigments found are R-phycoerythrin and chlorophyll A and D. We can find red algae in warm areas. We can find red algae in warm areas of water bodies. They can be found in shallow regions and depths of the ocean. Mostly multicellular organisms with having complex body structure. The food is stored in the form of Floridian starch, and Floridian starch has a similar structure to amylopectin and glycogen. Reproduction is vegetative reproduction is through fragmentation, asexual is through non-motile spores, sexual is through non-motile gametes which fuse, and it is mostly eugamous followed by complex developmental stages. Example are porifera, Polysiphonia, Gracilaria and Gelidium. With this we finish the algae topic. Next is the next division in the kingdom of Plantae is the division of Bryophyta. Bryophyta, right? It's Bryophyta. Let's see the structure first. Structure of the red algae. Here also we see a frond, which we call it as frond. Red algae, this is porifera. This is Polysiphonia. Polysiphonia, if we see, it is kind of like a land tree branch. In this diagram at least, it's like a dried or it's like a wilted branch or something like that. An old branch. Polysiphonia is kind of like it is not in our normal daily life we see such a structure, so we can't describe it. Porifera is like porifera. Then coming to bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shades in the hills. Basically, wait, let's see the table. I forgot the table. Okay, chlorophyce, common name is green algae. Pigments, chlorophyll A and B. Starch, swordfoot, starch, cell wall cellulose. Flagellar number and position of insertion, 2 and 8 equal and apical. Okay, fine, this I will write 2, 8 equal apical. It can be 2, 8, 2 hyphen 8 is a position, number of positions. 2 to 8 flagella will be there. Basically 2 to 8 flagella will be there. They will be equal and there will be apical. There doesn't seem to be a space here. I'll write it here. Flagella number from 2 to 8 and are apical. And equal. This is in spores and gametes. This is the next is habitat is freshwater, brackish water, and salt water. Here, in case of 505C, freshwater is rare, brackish water and salt water. Freshwater some, brackish water and salt water most. Marine it is most. 505C, brown algae, chlorophyll A and C, fucoxanthin, xanthophylls, carotenoids. Stored food is mannitol or mannitol laminarin. Here it is cellulose and pectose, gamma pectose. Here we have cellulose and algin. Algin is a gelatinous covering, and here it is unequal and lateral pyriform. I will not write pyriform here, it doesn't match. Habitat, yes, as I said before. Rhodophyceae, red algae, chlorophyll A, D, phycoerythrin. Stored food is Floridian starch. Cell wall is cellulose, pectin, and polysulfate esters. Here we saw cell wall, they didn't give anything about the cell wall. Cell wall is made of cellulose, pectin, and polysulfate esters. I will write that as well. Cell wall made of cellulose, pectin, polysulfate esters. Polysulfate esters. Bryophytes include the various mosses, and okay, fine, they are dividing the bryophytes in the first sentence itself. They are dividing bryophytes into mosses and liverworts that are found, they are giving the habitat which is growing in moist shaded areas in the hills, grows in the moist shaded areas, that is to in the hills. There should be moist habitat, they are growing in the hills, shaded areas. I'll see the structures later. Bryophytes are also called amphibians of the plant kingdom because these plants can live in the soil but are dependent on water for sexual reproduction. They're called amphibians of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. This statement is important. Bryophytes are called amphibians of the plant kingdom because these plants can live in soil but dependent on water for sexual reproduction. They live in soil only, but they depend on water for sexual reproduction. These occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks / soil. These bryophytes, they drill into the rocks, and they are one of the agents which helped in converting the rocks during the starting formation days of earth. The earth used to be filled with rocks, and during that time one of the factor which broke the rocks into soil was the plants which are bryophytes, which drill into the rocks and break them into two parts. The plant body of bryophytes is more differentiated than that of algae. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect. The plant body of bryophytes is more differentiated than that of algae. Fine, it is thallus-like only, but it is a little more differentiated compared to algae. It can be prostrate or erect, meaning it can be like lying down, or it can be like erect. It can be both. And attached to the substratum by unicellular or multicellular rhizoids. These rhizoids are false roots. They lack true roots. They lack true differentiation, but they have a little more body differentiation compared to thallophytes. In such a case, rhizoids are there, which are like true roots, but are actually false roots. They lack true roots, stem or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. Okay, the plant body is haploid. It produces gametes, hence is called a gametophyte. The main plant body is haploid and the plant body produces gametes. So in such a case, we call it as a gametophyte. The sex organs in bryophytes are multicellular. The sex organs are not unicellular but multicellular. The male sex organ is called the antheridium. They produce biflagellate antherozoids. The antheridium is a male multicellular sex organ and they produce multicellular antherozoids. The female sex organ called archegonium is flask shaped and produces a single egg. Basically, antheridium produces biflagellate antherozoids. Female sex organ is called archegonium and is flask shaped and produces a single egg. They gave three pieces of information here. One, antheridium is a male sex organ and it produces biflagellate antherozoids. Second information, the female sex organ is archegonium and is flask shaped. Third information, archegonium only produces single egg. The antherozoids are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce a zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called sporophyte. This sporophyte is not free living, but attached to the photosynthetic gametophyte and derives nourishment from it. Basically, after the description of sex organs, they gave 3 to 4 pieces of information. The first information is that the antherozoids are released into water. Okay, fine. Basically, the first information, how the fusion happens. The antherozoids are released into the water and reach the archegonium. In the archegonium, fusion of the antherozoids and the egg cell happen and a zygote is produced. This is the first information. The second information is that after this zygote is produced, it does not immediately undergo reductional division and it forms a multicellular body called sporophyte. This is the second information. Zygote is formed. This zygote doesn't undergo reductional division but forms a multicellular body called sporophyte. And the third information is that this sporophyte is not free living. It is not a free living body. It is developed attached to the photosynthetic gametophyte. The gametophyte is photosynthetic. The sporophyte is attached to the gametophyte and derives nourishment. Its food reserves, food needs, all other nutrient needs it takes from the gametophyte. Some cells of the sporophyte undergo reduction division, meiosis, to produce haploid spores. Basically, some cells of the sporophyte undergo reduction division, that is meiosis. Some cells of the sporophyte undergo reduction division. Okay, they're saying that this sporophyte is actually 2N diploid, because zygote is the one that is forming this sporophyte, and all the cells are diploid, that is of the sporophyte. The gametophyte is haploid, but the sporophyte is diploid. Some of the cells of this diploid sporophyte undergo meiosis to produce haploid spores. These spores germinate to produce the gametophyte. Okay, the spores are there which disperse, which then germinate, acting like seeds, to once again produce the gametophyte. Bryophytes in general are of little economic importance. Basically, bryophytes are basically there. I can't say, but they are basically useless. That is what little economic importance meaning. But some mosses provide food for herbaceous mammals, birds, and other animals. Okay, but some of the mosses provide food for herbaceous mammals, basically cows or sheep, goat, etc., birds and animals as well, other animals. Species of sphagnum, a moss, provide peat that have long been used as a fuel. Sphagnum is a species of a moss which provide a peat, PEAT, that is used as fuel and as a packing material for transshipment of living materials because of their capacity to hold water. They are basically used as fuel, and they can be used as packing material for shipment of living materials because their capability to hold water. Hydrocolloids, we saw this in case of algin and carrageen, produced respectively by brown and red algae, which are commercially produced or used as hydrocolloids, water holding substances. Here also peat, produced by sphagnum species, is used as fuel, as well as it is used in shipment or transport of living materials because of its capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and are great ecological importance. They decompose rocks, making substrate suitable for growth of higher plants. As I said before, during the first period of Earth, there were only rocks, and during such a time, mosses and lichens were the first ones to decompose these rocks and making soil which is suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. Bryophytes are divided into liverworts and mosses. This is the first main paragraph under bryophytes. Next comes liverworts and mosses, followed by pteridophytes. First, I will do with this one first. These are the Markantia, which is the liverwort, then followed by C is funaria, gametophyte and sporophyte. Sphagnum, a gametophyte. This is basically sphagnum. This is what produces the peat, which is used as fuel and transport shipment. This is the funaria, which shows this is the gametophyte stage. This is N. This is the ploidy of the gametophyte stage is N. This is the sporophyte, which is formed from the zygote. This is 2N. Capsule, which produces spores which are produced these pores are N because it is undergoing meiosis. The cells are undergoing meiosis to produce the spores, and these spores again germinate to give a, a sporophyte stage. I mean, no, a gametophyte stage, which again gives rise to a gametophyte, I mean a sporophyte, and this cycle continues. And this is a sphagnum. Here they only showed the gametophyte stage here. This is only the gametophyte stage. Okay, fine. Let's see the first information, the information under bryophytes. The first information is divided into mosses and liverworts. Divided into one mosses and two liverworts. Liverworts. Next, grow in shaded, moist and hilly areas. That is the second information. And the third information is bryophytes are called the amphibians of the plant kingdom. Called amphibians of the plant kingdom because the plants are on soil but dependent on water for reproduction. That is why they are called amphibians of the plant kingdom. They depend on water for reproduction. The fourth information is occur in damp, humid and shaded localities. They play important role in suction of bare rocks. I'll write it later. The plant body of bryophyte is differentiated into that of algae. Than that of algae, it can be prostrate or erect. The plant body is more differentiated than that of algae. The body can be erect or prostrate. The body can have unicellular or multicellular rhizoids. This is I think the fifth information. Then they lack true roots, stem or leaves. They possess root-like, leaf-like or stem-like structures. This is the fifth information. And one of the main information followed by that is haploid sporophyte stage. They lack true root, stem and leaf. Main plant body is Haploid. Main plant body is haploid. This is an important point. Main plant body is haploid. It produces gametes, so called gametophyte. Then I think they give information about the sex organs. Okay. Male antheridium, they produce biflagellate antherozoids. They produce biflagellate antherozoids. Female, it is archegonium, produce a single egg. The antherozoids are released into the water, hence they come in contact with archegonium. They produce biflagellate antherozoids. The female sex organ is called archegonium. Is flash shaped. Yes, they give flash shaped. And they give process of fusion of the gametes. The gametes, male will come in contact with female with archegonium, and fuse with egg to form a zygote. Zygote doesn't immediately get reduced, but forms a multicellular structure called sporophyte. The cells of the sporophyte undergo meiosis to form haploid spores. Spores germinate to give gametophyte. The sporophyte is dependent on the gametophyte for nourishment. Okay, this is for the general features of the mosses. I mean, not mosses, the bryophytes. Next will come the uses, but I will write the basic flow chart for the reproduction once. A reproduction flow chart. Which is basically anthrazoid, flagellate or motile. It was anthrazoid, biflagellate anthrazoids. Anthrazoids, male gamete, motile gamete, fuses with egg to give zygote to give sporophyte, then again differentiate to give spores again differentiate to give gametophyte, then gametophyte is the one that gives egg and anthrazoid. And depend on water for reproduction. This is the basic reproduction cycle. Then we will go for uses of bryophytes. The first use is not much economic importance but great ecological importance. Economic meaning commercial importance, but great ecological, breaking rocks, early phase of earth importance. Food for herbaceous mammals, birds. I think they gave insects, no, and other animals. Third, sphagnum, species of sphagnum gives peat used for fuel, used in transport of living materials. Because of the capacity to hold water. Mosses, along with the lichens, are the first organized organisms to colonize rocks and hence are of great ecological importance. The first point I'm repeating again: mosses and lichens are the first organisms to colonize rocks, so they have great ecological importance. And they form dense mats on soil, so they reduce soil erosion. I'll write it here itself. Form dense mats on soil. This is mosses. Prevent soil erosion. Yes, now that with that we have finished the main the general criteria of mosses. I'm not mosses, bryophytes. Let's see it again. Bryophytes are divided into mosses and liverworts. Grow in shaded, moist and hilly areas, called the amphibians of the plant kingdom because they survive on land but depend on water for like reproduction. The plant body is more differentiated than algae. The body can be erect or prostrate. The body may have unicellular, multicellular rhizoids acting as roots. They lack true root and stem and leaf. Main body is haploid and it is called a gametophyte because it produces gametes. Male gametes are, and male sex organ is antheridium and female sex organ is archegonium. The male sex organ, they produce biflagellate antherozoids. The female sex organ, archegonium, is a flask-shaped structure and produces a single egg cell. Antheridium, it is a biflagellate antherozoids. They are motile. And the gametes, male will come in contact with archegonium and fuse with egg to form a zygote. Zygote doesn't immediately undergo reduction division but forms a multicellular structure of sporophytes. Sporophyte cells divide meiotically to give haploid spores, which haploid spores germinate to give gametophyte. The sporophyte is dependent on the gametophyte for nourishment. It does not free living. Reproduction flowchart. Then uses: first use, no economical importance, not much economical importance, but great ecological importance. Food for herbaceous mammals, birds and other animals. Sphagnum gives peat, which is used for transshipment material and as a fuel because it has high water holding capacity. Mosses and lichens are the first organized to colonize rocks, to giving suitable soil for the growth of higher plants, and they prevent soil erosion by forming dense mats on the soil. The next is liverworts. liverworts. It's basically the same word. Liverworts under bryophytes. Under bryophytes we have liverworts. Basically, let's see. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. Example is Markantia. The thallus is dorsiventrally and closely pressed to the substrate. Basically, it is dorsiventrally flattened and closely pressed to the substrate. It is pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gemmae. Singular is gemma. Gemmae are green, multicellular asexual buds which develop into small receptacles called gemmae cups located on the thalli. The gemmae become detached from the parent body and germinate to form new individuals. The gemmae basically detach from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Basically, organs are produced on either the same or the different thalli bodies. The sporophyte is differentiated into a foot, seta, and a capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Okay, let's read that again. The first point is basically the habitat moist, shady, such as banks of a stream, marshy ground, damp soil, bark of trees, and deep in the woods. Plant body is thalloid. The thalloid is flattened, dorsiventrally flattened, and is pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows of the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli. Basically, asexual reproduction in liverworts takes place by fragmentation of thallus. Liverworts takes place by fragmentation of thallus. Okay, it might be due to fragmentation of thallus or the formation of specialized structures called gemmae or gemmae cups. Gemmae are green multicellular asexual buds which develop into small receptacle called gemmae cups located in the thallus. The gemmae become detached from the parent body. This paragraph is about asexual reproduction, about gemmae cups formation, and the first paragraph is about how the habitat of the liverworts, which is around the bark of trees, deep in the woods, banks of a streams, marshy ground, and how the thallus is dorsiventrally flattened and closely pressed to the substrate, and how leafy members have leaf-like appendages in two rows to the stem-like structures, and the next paragraph is about asexual reproduction in the liverworts, which is by fragmentation of the thallus or by the formation of specialized structures called gemmae. Gemmae are green. Okay, this is about gemmae cups. First is fragmentation, asexual reproduction, fragmentation. The second is by gemmae. These gemmae are multicellular. They are multicellular asexual buds that are located on the thalli and which develop into small receptacles called gemmae cups located on the thallus. These multicellular asexual buds In the initial state, gemmae are buds. These asexual buds later develop into small receptacles. These receptacles are called gemmae cups, are located on the thallus. The gemmae becomes detached from the parent body and germinate to form new individuals. Basically, the initial stage is a multicellular asexual bud. This bud develops into a receptacle called gemmae cup. This gemmae cup detaches from the parent body and forms a new thallus. During sexual and next asexual reproduction paragraph information is completed. Sexual reproduction, male and female sex organs are produced either on the same or different thallus. It can be bisexual or unisexual. The sporophyte is differentiated into after fusion. Sporophyte is formed, and this sporophyte is differentiated into a foot, seta, and capsule. Basically, here we can see it is not clearly given in the Markantia state, which is just showing the sex organs. It is not showing the sporophyte stage, but here in case of moss, we can see this long stalk is called seta. This is the capsule, and this is the foot, is basically this one itself. And here this is the structure of Markantia, which is a liverwort. These structures are the gemmae cups, rhizoids, root-like structures. The leafy structure having notches, and this is the archegoniophore. Archegoniophore meaning female sex organ. This is the antheridiophore, male sex organ. These are the gemmae cups here as well. These are the rhizoids. These organs are either both are produced on the same organism or on different thallus bodies. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. Same as what we said, read in the bryophytes. It is actually bryophyte only. Under bryophyte only, we are reading different division of the liverworts, which they gave to be under 2 paragraphs. Okay, fine. Let's write them. First is habitat. Grow in damp, moist regions like banks of streams, bark of trees, then deep in woods, etc. I don't think in here they mentioned stones here. They didn't mention stones here. Yes, they mentioned marshy ground, so it might be considered actually marshy ground, etc. Then they grow in regions like that. Then the next information was about how the like dorsiventrally suppressed. The body is dorsiventrally flat and is pressed to the substrate. That is the about leafy as well. For leafy individuals, two rows of leafy appendages on stem-like structure. Then the next information is about reproduction. Reproduction asexual reproduction or vegetatively, A through fragmentation and B is through gemme formation. Gemme formation is basically a cycle of gemme is basically asexual multicellular buds. These develop into gemme cups, receptacles. Actually, it is better, I think, if I write sentence wait, sir. Gemme later develop into asexual receptacles called gemme cups. Gemme cups detach from the body to form individual organisms or germinate into individual organisms. This is asexual reproduction. Then we will go to sexual reproduction. Sexual reproduction is through antheridium, male, and archegonium, female, which are formed on the actually either there I missed one information, that is either both or individual sex organs are formed on the body. Okay, yes, then these are and the process I told in the bryophytes itself, like it is under bryophyte, so it's the same process. Then after that, a sporophyte stage containing a foot, seta, and capsule are formed. Haploid spores produced, which germinate into new gametophytes or new organisms slash gametophytes slash plant body. And this is the total amount of information given here, I guess. Sporophyte is okay, yes, after meiosis, spores within the capsule, these spores are not okay, yes, free living gametophytes. And example are one and only Markantia. This is the end of liverworts, but not the end of bryophytes. Then actually let's do a revision. Let's go back to blue color. No, let's go back to green itself. Green for mosses. Let's do a quick revision of the liverworts. Grow in damp, moist regions like banks of a stream, banks of bark of a tree, deep in woods, marshy areas. The body is dorsiventrally flattened and oppressed to the substrate. For leafy individuals, there are leafy appendages on the stem-like structure. Reproduction, asexual reproduction, vegetative reproduction. It is one is through fragmentation and other is through gemme formation. Gemme is a multicellular asexual bud which later turns into small receptacles of gemme cups. These gemme cups detach from the main body to germinate new individual organisms. And sexual reproduction is through during sexual reproduction, the antheridium and archegonium sex organs are formed. Either both or individual sex organs are formed on the body. And sporophyte stage containing a foot, seta and capsule are formed. Haploid spores produced which germinate into new organisms called gametophyte, which is example is Markantia. Basically nothing much actually. It took time for writing, that's all. Now mosses. The predominant state of all the life cycle of a moss is the gametophyte, which consists of two stages. The predominant stage of life cycle of a moss is gametophyte, which consists of two. Basically saying first information is the dominant stage of the life cycle of a moss is the gametophyte. But there are total of two stages. The first stage is the protonema stage, which develops directly from a spore. Spore to protonema. The first stage is developed from a spore. This is called protonema stage. It is a creeping, green, branched and frequently filamentous stage. It is a creeping, meaning prostrate. It is green in color. It is branched and it is frequently filamentous stage. The second stage is leafy stage, which is developed from the secondary protonema as a lateral bud. Basically, this is the ground, a spore is there. A spore will germinate to give a creeping green structure which will have branches. It is filamentous and this is the protonema stage. Protonema stage, and from this buds arise. These buds develop into a leafy stage, and this is the diagram. It's not really neat, but this is the sporophyte stage, this is the leafy stage, this is the protonema stage. Directly from spore is called protonema to leafy stage. The second stage, lateral bud developing into a leafy stage, which develops from the secondary protonema as a lateral bud. A secondary protonema, it's directly not from a secondary protonema, it will develop as a lateral bud. They consist of upright slender axis bearing spirally arranged leaf like this. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Basically, that's why you call it as a gametophyte. Vegetative reproduction in mosses is by fragmentation, and the budding is the secondary in the vegetative reproduction in mosses is by fragmentation, but the vegetative reproduction in protonema stage and the secondary protonema stage is budding, a lateral bud which will give rise to a new organism of the leafy stage. In sexual reproduction, the sex organs are Antheridia and Orchigonia are produced at the apex of leafy shoots. Okay, these organs are produced at the apex of the leafy shoots. After fertilization, the zygotes develop into a sporophyte, okay, consisting of a foot, seta and capsule, same thing repeated. The sporophyte in mosses is more elaborate than liverworts. The sporophyte in mosses is more elaborate than liverworts. The capsule contains spores. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Okay, fine. Let's read the information again. Basically, the predominant stage of the life cycle of a moss is the gametophyte, which consists. Basically, the predominant stage is gametophyte only. It's not the sporophyte. Sporophyte is not a free living stage, and it is dependent on the gametophyte. And this gametophyte is again divided into two stages. The first stage is the protonema stage, and this protonema is a creeping, green, branched and filamentous stage. And the second stage is a leafy stage, which is developed basically from a branch, a secondary protonema, which developed from a secondary protonema. Protonema is the leafing, crawling or creeping structure. This is the protonema stage, which is developed from a spore. And after that, lateral buds on the protonema arise, and these buds develop into the second stage, the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender, like they're describing the leafy stage. They consist of the upright, slender axis bearing spirally arranged leaves. This is an upright axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. Okay, they are attached to the soil through multicellular and branched rhizoids. This stage bears sex organs. Basically, they are saying they are attached to the soil through multicellular and branched rhizoids. Okay, this stage bears the sex organs. They are talking about the leafy stage. Leafy stage bears the sex organs. Vegetative reproduction is in mosses is by fragmentation, and in secondary protonema is by budding. In sexual reproduction, the sex organs Antheridia and Orchigonia are produced at the apex of the leafy shoots. These sex organs Antheridia and Orchigonia are produced at the apex of the leafy shoot. After fertilization, the zygote develops into a sporophyte. After fertilization, like as basically described before, a zygote develops into a sporophyte consisting of a foot, seta and capsule. The sporophyte in mosses is more elaborate than in liverworts. The capsule contains spores. Spores are elaborate, meaning more detailed or something like that. Or more complex also, we can say more detailed, more complex, or more, it's detailed itself. Capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. These mosses have developed an elaborate mechanism of spore dispersal. The common examples of mosses are Funaria, Polytrichum, Sphagnum, etc. Okay, let's start writing. The first information, the predominant stage of mosses is the gametophyte. The gametophyte is again divided into two stages of a protonema stage, B, leafy stage. Protonema stage is basically a creeping green branched and often filamentous structure is the protonema stage. From the bud, the lateral buds on the protonema stage give rise to lateral buds on the protonema stage give rise to the leafy stage, the second stage. It's basically from the secondary protonema they mentioned, but I just mentioned protonema here. And leafy stage description: a stage of upright, axially meaning erect, axially arranged or spirally arranged leaves. A stage of upright, axially arranged or spirally arranged leaves. Presence of multicellular rhizoids. The leafy stage bears sex organs. Basically Antheridia and Archegonia, and the fusion of gametes leads to zygote formation. Zygote forms sporophyte. Sporophyte undergoes, some cells of sporophytes undergoes meiosis to produce spores. Spores germinate into the new plant body. I think this is pretty much the information they gave, except the examples. Predominant stage is gametophyte, and 2 stages of protonema and leafy, and protonema is directly formed from a spore. Formed directly from a spore. Okay. It is creeping green branched with frequently filamentous stage. The second stage is the leafy stage. We develop from the secondary protonema as the lateral stage. As the lateral bud. They consist of upright slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. I missed vegetative reproduction. Vegetative reproduction in mosses is by fragmentation, and for protonema it is via budding. This is vegetative reproduction. Zygote develops into a sporophyte, consists of a foot set and capsule. The sporophyte in mosses is more elaborate than the zygote develops into a sporophyte, consists of foot set and capsule, more elaborate. The sporophyte of mosses is more elaborate than liverworts. Is more elaborate than liverworts. The sporophyte of mosses is more elaborate than liverworts. The mechanism of dispersal of spores is also more elaborate than mosses. Okay, fine. This is basically all of the information, and the examples are Funaria polytrichum and sphagnum. Etc. This is with Funaria. I mean, not Funaria. This is with the mosses. This is with the liverworts. This is with the bryophytes. Bryophytes is therefore completed. But let's see mosses once more. Predominant stage of mosses is the gametophyte. The gametophyte is again divided into two stages of protonema and leafy stage. Protonema is directly or stage directly formed directly from a spore, and it is a creeping green branched and often filamentous structure. The lateral buds on the protonema stage give rise to the leafy stage. Leafy stage, a stage of upright, axially arranged or spirally arranged leaves, presence of multicellular rhizoids. The leafy stage bears sex organs, antheridia and archegonia. It's actually antheridia 4 and archegonia 4. The fusion of gametes leads to zygote formation. Zygote forms sporophyte. Sporophyte produces spores. Spores germinate into the new plant. Vegetative reproduction in mosses is by fragmentation, and for protonema it is via budding. The sporophyte of mosses is more elaborate than liverworts. The mechanism of dispersal of spores is also more elaborate than mosses. Example for the mosses is Funaria polytrichum and sphagnum. Sphagnum uses of sphagnum is production of peat. Peat is used in fuel, used as fuel, and it is also used in transport of living materials, like due to its high water holding capacity. That's all for the bryophytes. Then the next division under the plant kingdom is pteridophytes. The color, I think red for roses. Rose is a pteridophyte. At least some species, I think so, right? Is rose a pteridophyte? No, rose is not. Okay. Okay, rose is not a pteridophyte. Okay, sorry. But still, let's use red itself. Pteridophyte or pteridophyta. First vascular plants, meaning they contain xylem and phloem. The first plants to ever have xylem and phloem are the pteridophytes. Okay, let's see. Pteridophytes again divided into, wait, it's around 2 paragraphs. It's not much information. Fine. Pteridophytes, the pteridophytes include horsetails and ferns. Okay, there's again one more division under the pteridophytes, which is horsetails and ferns. When we saw division under Thallophyta, it was Chlorophyaceae, Phaeophyaceae and Rhodophyaceae. Under like Bryophyta, we saw it was mosses and liverworts. Under liverworts, it is leafy stage and leafy stage and sporophyte under the liverworts, and under the mosses we have the gametophyte, sporophyte, and under the gametophyte we have protonema and leafy stage. So pteridophytes we have horsetails and fern. This is kind of like a division under the bryophytes, which is liverworts and mosses. Pteridophytes are also divided into horsetails and ferns. Horsetails. Pteridophytes are used for medicinal purposes and as soil binders. Okay. Soil binders meaning we saw mosses, mosses are spread on mats, which we saw in the uses of mosses. Not uses of mosses, uses of bryophytes, which mosses are spread on the ground as mats, which tightly bind the soil and prevent soil erosion, reducing the impact of the rain on the soil. Here as well, they are used. Pteridophytes uses are used for medicinal purposes and used as soil binders. They are frequently grown as ornamentals evolutionarily. Okay, they are frequently grown as ornamentals, and the next is evolutionarily. They are first terrestrial plants to possess vascular tissue. The first vascular plants to possess xylem and phloem. You shall study more about the tissues in chapter 6. Okay. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. Basically, the pteridophytes are found in cool, damp, shady places, like similar to how the environment of bryophytes and Chlorophyaceae. The algae was under in water aquatic conditions, but in bryophytes we saw it was in damp and cool conditions. Here as well, pteridophytes also, it is also in cool, damp, shady places, but some may also be in sandy soil conditions like desert conditions. You may recall that in bryophytes, the dominant phase in the life cycle is a gametophytic plant body. Basically, the plant body is haploid, only half number of chromosomes will be present. So it is called a gamete producing body, is called a gametophytic body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem and leaves. Pteridophytes are not only the first vascular plants, but pteridophytes are the first plants to have complete body differentiation into true leaves, true stem and true roots. these organs, okay, the point is they have true root, true leaf and true stem, and their body is sporophytic, exactly different from bryophytes which had a gametophytic plant body. And these organs possess well differentiated, they gave a figure 3.3, let's see the figure 3.3. These are all pteridophytes. Pteridophytes, A is Selaginella, B is Equisetum, C is a fern, and D is Salvinia. Fine, these are just names. These organs possess well differentiated vascular tissues. Okay, they possess well differentiated vascular tissue. The leaves in Pteridophyta are small microphylls as in Selaginella, or large macrophylls as in ferns. Basically, the leaves may be small microphylls, phyll meaning leaf, micro meaning small, like Pteridophyta, as in Selaginella. Selaginella is the first one, or the next is as in ferns, they said. Here, if we see the leaves, they are very small. This is a leaf, this is a microphyll, small leaf. Now let's see ferns, the macroscope, I mean not macroscope but macrophyll. Here if we see, if we zoom in, the leaves are large leaves. Macrophyll, large leaves, fill being leaf, macro being large leaf. This is the example for fern. And this is the example for Selaginella. This is Equisetum, Strobelus cones, we will indeed read about them. Okay, the information is saying the leaves can be differentiated into small microphylls as in Selaginella, or large macrophylls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. Basically, here the body itself, we saw it is a sporophytic body, meaning the body is diploid and it is bearing spores, and the sporophytes bear sporangia. Basically, these sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. Let's see what this means. Basically, let's see the statement itself. I'm not sure if the sporophyte is referring to the plant or a special organ. Here, or this statement is from under pteridophytes. here the term does the term sporophyte refer to the plant and sporangia same as sporophyll? No, wait. And are different kind of spore producing leaves? This is my doubt. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophyll. The sporophyte refer to plant itself? Yes. Pteridophytes, the main plant body is sporophyte. It is diploid to end and dominant in the life cycle. In case of bryophyte, we saw that the predominant or dominant stage was gametophyte, which was the end, a single ploid. So the sporophyte, the leafy plant you see in ferns and the other pteridophyte. Okay, the sporophyte, the leafy plant you see in ferns and other pteridophyte. This is the plant itself, and it produces spores via meiosis. Okay, are sporangia and sporophylls the same? No, they are not the same. Sporangia structures usually small sacs or capsules that produce and contain spores. Okay, basically sporophylls are leaves, and basically sporophylls are leaves, or we can say leaves bearing sporangia. Sporangia are structures which produce and contain spores. The leaves having this kind of sporangia are called sporophylls. What does subtended by sporophylls mean? Positioned beneath or supported by. So sporangia are supported on or attached to sporophylls, special spore bearing leaves. Are sporophylls different from normal leaves? Yes, normal leaves in pteridophytes do photosynthesis. Okay, sporophylls do not do reproduction, they bear only sporangia. In pteridophytes like Selaginella and ferns, sporophylls may look similar to normal leaves or be very distinct and grounded into structure like strobe-like cones. Okay, visual analogy. Okay, right. Think of sporophyll, branch, and sporangia, fruit hanging from it. Okay, both are of the plant but have different roles. Basically, sporophylls are leaves which bear sporangia. Sporangia are the spore producing structures. And here this sporophylls may be similar to normal leaves in case of Selaginella and ferns, or in case of Equisetum we see special cones here. This cone, the strobilus, it may form these special cones. Okay. Okay, then let's continue. In some cases, sporophylls may form distinct compact structures called strobili or cones, Selaginella and Equisetum. Okay. The sporangia produces spores by meiosis in the spore mother cell. Okay, basically sporangia is formed from the plant cell. Plant cell are diploid because it's the main dominant stage of the sporophyte. The pteridophyte plant is diploid or sporophyte. So in such a case, the plant cells will specialize to form spore mother cells. Spore mother cell are diploid, and spore mother cell undergo meiosis to produce sporangia or spores. Actually, not sporangia, spores. The sporangia produce spores by meiosis in spore mother cell. The sporangia produce spores by meiosis in spore mother cell. The sporangia produce spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. Let's read the statement again. The spores germinate to give rise to inconspicuous, small, basically what the germs germinate into, not germs, the spores germinate into is, the spores germinate to give rise to inconspicuous, meaning not seen, like not very visible, small but multicellular, they're multicellular, free living, mostly photosynthetic thalloid gametophytes called prothallus. Basically, in case of bryophytes, we saw that the gametophyte is a predominant stage and gametophyte will give rise to the sporophyte, and in that case the sporophyte is not free living, it is dependent on the gametophyte. But here they are saying that the plants will give rise to special, the spores will give rise to special gametophytes, or basically the spores will give rise to small inconspicuous gametophytes. Yes, gametophytes. These gametophytes are multicellular and they are free living, and these gametophytes are called prothallus. Yes, that is what. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and need for water for fertilization, basically they are saying these gametophytes require cool, damp, shady places to grow. And because of that requirement and the need for water for fertilization, they need water for fertilization. These restrictions, due to the such restrictions, the spread of living pteridophytes is limited and restricted to narrow geographical regions. Only in specific geographic regions we can find pteridophytes. Otherwise it is limited because of water, special conditions, everything is needed. The gametophyte bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids, the male gametes released from antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium results in the formation of zygote. Everything repeat of the before processes. Zygote thereafter, and here it is a different one. Just a minute. Okay. Okay, fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte. Okay, zygote thereafter produces a multicellular, well-differentiated sporophyte. Okay, okay, this zygote, like after formation of zygote, this zygote will transform into a well-differentiated sporophyte, which is dominant phase of the pteridophytes. Basically the cycle, I'll write it later. In majority of the pteridophytes, all the spores of similar kinds, such as all the spores are of similar kinds. Such plants are called homosporous. Okay, they are introducing a new concept here, which was not in the previous topics. That is spore kind, which was in algae, but not in bryophytes. Homosporous. If majority of the pteridophytes, in majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro and microspores, are known as heterosporous. Basically, homosporous is not within many genera or many kind of plants. It is in a single plant, where a plant does not differentiate its spores and produce single spores, single kind of spores. Then it is called homosporous. But when in Selaginella and Salvinia, these produce two kinds of spores. One is the macro and one is the microspores. These are known as heterosporous. And in the majority of the pteridophytes, all the spores are similar kind. Basically, homosporous plants are more in the pteridophytes compared to the heterosporous. The megaspores and microspores germinate to give rise to female and male gametophytes. Basically, megaspore will germinate to give rise to a female gametophyte, and microspore will give rise to a male gametophyte, respectively. The female gametophytes in these plants are retained on the parent sporophyte. Okay, they are introducing one more specialty here. It's not a concept, it's a specialty. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of the young zygotes into young embryos take place within the female gametophyte. This event is a precursor to the seed habitat, considered an important step in evolution. Okay, basically they introduce a specialty of the pteridophytes here. A specialty is in heterosporous plants, the female gametophyte in the plant, like after fertilization, it will stay on the plant itself. It depends on the main sporophyte plant. It's a gametophyte, and this special female prothallus will stay on the parent itself, the sporophyte plant, and it will be retained there for variable periods until the fertilization has happened and zygote develops into a young embryo. This event, it is similar to how the modern angiosperms, they produce seeds. It is similar to how that happens. That is why it's a precursor to the seed habit, considered an important step in evolution of the plants. That is what they mean here. The pteridophytes are further classified into four classes of Psilopsida. Example is Xylotum. Then Lycopsida. Example is Selaginella, Lycopodium. Sphenopsida. Example is Equisetum. And Pteropsida, Rhiopteris, Pteris, and Adentium. This is the pteridophytes. Okay, let's see the information once more. The pteridophytes include horsetail and ferns. First information, division of the pteridophytes is divided into horsetails and ferns. Pteridophytes are uses of pteridophytes are medicinal purposes, soil binders, prevent soil erosion. Second information completed. Third information, They are frequently grown. Okay, this is also uses only, so second information only. Uses are medicinal purposes, act as soil binders, and as used as grown for ornamentals. That is the second information. And third, they are the evolutionarily they are the first terrestrial plants to possess vascular tissues, that is xylem and phloem. And the fourth information, these pteridophytes habitat. The pteridophytes are found in cool, damp, shady places or even in desert or soil conditions, sandy soil conditions. You may recall that in bryophyte, okay, basically they are saying the plant body, the fifth information is the plant body of pteridophytes is sporophyte. It is a diploid stage. And the sixth information is the bodies is well differentiated, having a true root, true stem, true leaf, and these organs possess well differentiated vascular tissues. And the seventh information, differentiation or variation in leaves, which is divided into small microphyls, and in case of Selaginella, or large macrophyls as in ferns. Okay, the sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. Basically, the eighth information is sporophytes. Basically, these plants, they have special leaves which bear sporangia. Sporangia are structures which produce spores, and these leaves which bear these sporangia are called sporophylls. And in some cases, in case of normally ferns, if we see, it is not differentiated. These these sporophylls and normal leaves are not differentiated, but in case of equisetum, if we see, these sporophylls are transformed into a compact structure called a strobilus, which is a cone-like structure. And the sporangia, the ninth or tenth information is like sporangia has sporangia mother cells. Sporangia mother cells are the plant cells itself. Plant has a sporophyte dominant stage, and it has a sporophyte dominant stage, so it has diploid cells, and sporophyll mother cells are also diploid, and these sporophyll mother cells will undergo meiosis to produce spores. And these spores, next information, 11th information, is these spores germinate to produce a small inconspicuous multicellular gametophyte. It's a free living gametophyte in most cases. It is photosynthetic thalloid gametophytes called prothallus. These gametophytes require, they are giving a condition here, for this formation of these gametophytes, this prothallus, they need cool, damp and shady conditions, and they need because of this, like they also need water for fertilization, for the fertilization of the gametophyte. So due to such restrictions, the pteridophytes are limited to certain geographical places. That is the 11th information. The 12th information, the gametophyte bears both either male or female sex organs called antheridia and archegonia for respectively. Wait, I have a doubt. In the prothallus / gametophyte, do the prothallus produce both male antheridium and archegonia force on the same prothallus Okay, it seems does a single prothallus produce both antheridia and archegonia? Yes, in most ferns, a single prothallus is bisexual. Okay, it is also called monoecious and produces antheridia male and female sex organs, and they have tiny heart-shaped or thalloid prothallus bearing both male and female. Okay, antheridia force is used in. Okay, it is not in pteridophytes, it is in bryophytes. Okay. Then I have one doubt. If a single prothallus is producing both the male and female sexual organs, what is the use in sexual reproduction? Sexual reproduction is for variation. So different species should be, or different plants should be introduced. But if both are in such a case, what is the use for sexual reproduction? I have one more doubt. Is this only in homosporous plants? In heterosporous plants, they clearly said that the male and female are different. If the prothallus is producing both the male and female sex organs, then what is the need for sexual reproduction? Is this bisexual prothallus only in homosporous plants? Yes, mostly homosporous. Example ferns equisetum produce only one type of spore, and the spore grows into a bisexual gametophyte which produces both antheridia and archegonia. So the same gametophyte can potentially produce both male and female gametes. Heterosporous pteridophytes, example Selanginella and Marsilia, produce two types of spores, micro and megaspores, and in microspores, as I said before, they grow into only male gametophytes. In megaspores, only female gametophytes. These gametophytes are unisexual and are often greatly reduced, sometimes even non-photosynthetic. If the prothallus is bisexual, what is the need for sexual reproduction? Here's the key point. Even though single prothallus can produce both sex organs, fertilization still involves the fusion of two gametes, sperm plus eggs, which brings genetic variation. Okay. To favor cross pollination, not selfing. Earlier than archegonia, sperms are but to bring different plants when us has both male and female organs, then when fertilization is still functionally cross pollination, but genetically self pollination, not pollination but fertilization. Or is it that even though both the organs are on the prothallus, there will be no self-fertilization due to outbreeding device slash mechanism? Yes, you're right. When both Antheridia and in terms of structures, they use outbreeding devices to favor cross-pollination, such as, okay, it's like malic acid to attract more cross. Then why is the prothallus bisexual at all? This is one of my questions. In low density populations, self-fertilization is better than no fertilization at all. Okay, I understand. Okay, it ensures reproductive, it ensures that at least a new plant is formed. Like, instead of waiting for someone that, or the new fertilization things that might or might not come, it is better to have a kind of a backup plan when there is no attraction of other individuals like male or anthrazoids or maybe the egg. So in such a case, to assure itself, the prothallus will be bisexual. It is a flexible strategy, but can fall back on selfing if needed. Okay. Okay, thyroid glands are called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of anthrozoids, the male gametes, released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte which is dominant phase of pteridophytes. In majority of the, basically after fertilization, the gametophyte will germinate into a sporophyte. In majority of the pteridophytes, all the spores are similar kinds. Such plants are called homosporous. Genera like Selaginella and Salviniella, which produce two kinds of spores of macro and microspores, are known as heterospores. The megaspores and microspores germinate and give rise to female and male gametophytes. The female gametophyte in the plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos takes place within the female gametophyte. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes of Celopsida, Xylotum, Lycopsida, Selaginella lycopodium, Sphenopsida, Equisetum, and Pteropsida, Diopteris, Pteris, and Adiantum. Before writing, let's see the information once more. Pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes, soil binders, ornamentals. Evolutionary, they are the first terrestrial plants to possess vascular bundle or vascular tissue. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle in the gametophytic plant body is a gametophytic plant body. However, in the pteridophytes, the main plant body is a sporophyte, which is differentiated into a true root, stem, and leaves. These organs possess well differentiated vascular tissues. The leaves in pteridophyta are small microphylls as in Selaginella or large macrophylls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones, which in case of Selaginella and Equisetum, the sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living gametophytes, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow because of the specific restricted requirement and the need for water for fertilization. The spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids. The male gametes released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygote into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. Great. The pteridophytes are further classified into four classes: Celopsida, Xylotum, Lycopsida, Selaginella lycopodium, Sphenopsida, Equisetum, and Pteropsida, Diopteris, Pteris, and Adiantum. So let's write this. Let's see the structures here. They gave Selaginella, Equisetum, fern, and Salvinia. In Selaginella, we see microphylls, small leaves. We see in Selaginella heterosporous. It is a heterosporous condition. Yes, it's a heterosporous condition. It produces two different kind of spores through leaves, stem, and roots. and this is fern macrophyls, and fern doesn't have strobilus. If you see the fern, you don't see macrophyll, I mean not macrophyll, strobilus. You see that the sporophylls are not differentiated into special structures of the cones, and in equisetum you do see the strobilus, which is the special structure of the cone, and these are the nodes, internodes, branches, and the rhizome. This is the Salvinia. Salvinia also produces heterospores. It's a genera which produces heterospores, and I will end the class after completing the writing of this one. Pteridophytes, vascular plants. First information, divided into A, horsetails, B, ferns. Second, uses of pteridophytes. Uses of pteridophytes are: 1, used for medicinal purposes. 2, not used but act as soil binders therefore prevent soil erosion. 3, it's used as ornamental plants, decorations, and these are the uses of pteridophytes. Next information is, they are the first terrestrial plants to possess vascular system. Xylem and phloem. Next, found in cool, damp and shady places, or even in sandy soil conditions. Fine. Okay, next information. Dominant phase of plant life is the sporophyte stage, which is 2n, diploid. Next information, they possess well differentiated stem, leaf and roots, basically true organs, plant organs. And vascular tissue is spread through all of them. Spread through all of them. Next information is about the different variation in leaves. The leaves can be microsporophyll, microphyll, not microsporophylls, small leaves, example Selaginella, can be macrophylls, large leaves, example it is fern. Next information is the plants, sporophytes, have special leaves bearing sporangia, bearing sporangia, which is spore producing organ, or we can say organ. Are called the plants. The sporophytes have special leaf bearing sporangia, are called sporophyll. Plant sporophytes have special leaves bearing sporangia, spore producing organ are called sporophylls. And the next information, okay, yes, normally the sporophylls, actually not normally, the sporophylls may be identical to normal leaves or form distinct compact structures called strobilus / or cones. This is then what about the next information? Yes, the cones and the sporangia produce the sporangia produce spores from the spore mother cells SMC which divide meiotically. Next information, then the spores germinate together as to spores produced, actually the spores, I'll write just spores, then germinate to give rise to small inconspicuous multicellular gametophytes called the prothallus. Prothallus. Prothallus is nothing but the gametophyte, gamete producing body. These prothallus are thalloid and prothallus are thalloid and photosynthetic. Some cases it may not be. And extra information, the prothallus in case of homosporic plants bear both male and female sex organs. Bisexual prothallus. Although they prefer sexual reproduction, this is as an assurance in case there is no cross pollination, not cross fertilization happening. Turn into a box. It's not turning into a box. It's again not turning into a box. Finally. Okay, anyways, The prothallus are thalloid and photosynthetic. Let's see the information again. And these prothallus require cool, damp and shady places / environment, and the fertilization is depending on water. Due to these restrictions, the pteridophytes are generally found only in specific geographical regions. Okay, that is one information. Next, basically the same thing repeating again. Well differentiate sporophyte. After fertilization, antherazoid plus egg gives zygote. Zygote is developed into a mature sporophyte. The dominant plant phase. And next topic is, in majority of pteridophytes are homosporous they produce homosporous produce same kind of spore only, or only one kind of spore. But there are plants which produce two kinds of spores. That is a, or I'll write first kind, microspore give rise to male gametophyte. Megaspore give rise to female gametophyte. Examples for actually these plants producing two kinds of spores are called heterosporous. It became a ray. Wait. No, it needs to be a perfect box. Heterosporous, yes, anyways. There are plants which produce two kinds of spores. These are called heterosporous. Okay. Then example for these are I think Selaginella, genera of Selaginella, Salvinia, etc. These two genres, I think, yeah, these two genres which produce two kinds of spores, heterosporous. And one more mechanism here, the female speciality of female gametophyte. The female gametophyte is not detached from the parent body for an invariable amount of time till fertilization and until after the zygote formation. This speciality is important as it is one of the no, it is not one of the, it is the precursor to the evolution of seed habit. And one more main topic is the classes under pteridophytes is one Coelopsida, I think. Psilopsidum. Wait, spelling is Psilotum. Second, Lycopsida. Example, it is Salvinia and yeah, it is Selaginella and Lycopodium. The third class is Sphenopsida. Example, Equisetum. Four, Pteropsida. Example, it is three examples here: Dryopteris, Pteris, Adiantum. With this, we have completed pteridophytes. The next two topics are gymnosperms and angiosperms, which is around 3 pages, but I will not be doing it right now. I'll be doing it in my next class, but I will revise these classes once. The classes names are hard to remember. Classes under pteridophytes are Psilopsida, example Psilotum, Lycopsida, Selaginella and Lycopodium. I will underline them as well. Sphenopsida, Equisetum, Pteropsida, Dryopteris, Pteris and Adiantum. Psilopsida, it is Psilotum. Lycopsida, it is Lycopodium and Selaginella. Sphenopsida, it is Equisetum. Pteropsida, it is Dryopteris, Pteris and Adiantum. Okay then, good night. See you in the next video.